we talk about just briefly cell division. And there's two types of cell division in the human body. There's mitosis and meiosis. Be able to differentiate between the two. Meiosis is straight up gamete division. All right. So oocytes are eggs in females, sperm in males. All other division or all other cells, which are called somatic cells, will engage in mitosis. And usually a result of mitosis means you start with one parent cell that divides and it gives you two daughter cells, which will be identical. All right, so with gamete division, gametogenesis, that's a little bit different here. All right, so what we're going to see is we're going to see the sex cells, right, are male cells, the sperm, and the oocytes, the eggs. They're going to undergo this process of meiosis, and we're going to go through some of those processes here, except with a few different differences that you're going to notice if you're familiar with mitosis, and if you're familiar with meiosis a little bit, you're going to notice some of the differences there. All right, there we go. Why aren't you working? There we go. Okay, so couple finer points. With meiosis, you're going to start off with, all right, a parent cell. Now, diploid means you have two copies, right, of the same chromosome, meaning one comes from mom and one comes from dad. Or you have a maternal chromosome and a paternal chromosome. Right? So if you haven't taken genetics before, there are 46 chromosomes in humans. Right? So 23 come from mom and 23 come from dad. And so the gametes, those are going to be the oocytes, the eggs, and the sperm, right? They're going to be known as haploid, which means they will have half this number of 46, which will be 23. And we'll discuss how that's going to happen. But essentially what we're going to do is when you have fertilization, when you have union between the sperm and the oocyte will have 23 chromosomes. The sperm will have 23 chromosomes. They'll reunite to give you that diploid number, that 46. So that's essentially what we're trying to do. We're trying to create an organism, right, that is going to have the normal number of human chromosomes here. And so that process is going to be, right, meiosis, which we'll discuss here shortly. So you can see the differences between mitosis and meiosis, right? Mitosis, somatic cell, it's pretty much all body cells that are going to engage in mitosis if they're capable. Unfortunately, a neuron is amitotic, so it will not engage in mitosis there. Right? So meiosis, sexual cell division. At the end of mitosis, you wind up with two daughter cells, and they're going to look the same as the parent cell. So you learned about that in chapter 6, right? When you get the keratinocytes and the stratum basal, right? That cell, those basal cells there will divide. Right, one cell will stay in the stratum basal to divide again, and the other cell, the other daughter cell, will migrate into the stratum spinosum. That's an example of mitosis. Same cell, 46 chromosomes, identical to the parent there. Right, so keep in mind, you'll have a diploid number, 46 chromosomes. And this process we'll talk about here, crossing over is what gives us our genetic diversity. So again, you'll notice that you might look similar to mom and dad, but you don't look exactly like mom and dad. Okay, so that crossing over allows for this genetic diversity that we see across, right, the human population there. So with meiosis here, instead of having two daughter cells, you're going to have 4 daughter cells. But here's the catch. The 4 daughter cells are going to be haploid. They will have half the number of chromosomes. So that means they'll have 23 chromosomes instead of the 46. And so you will see crossing over, and I'll show you that shortly here when we get into the different phases of meiosis here, but you will get crossing over. And this term here, homologous, do y'all know what that means? Something's homologous. It's the same. Yeah. So you have heterogeneous and then you have homologous. Heterogeneous means there's a mixture, the diversity there. Homologous means the same. So what that statement there means, you're going to get, all right, crossing over of genetic material, because remember what I said, you get 1 chromosome from mom and 1 chromosome from dad. So what you're going to see is those identical chromosomes will just cross over. And so the maternal and paternal chromosomes will exchange with one another. And it does it randomly. There's no specific chromosomes that only cross over. It's just a random, it's kind of a cool event. They'll just randomly just exchange genetic material, right? And so again, that's why if you look around, everybody in this room looks relatively different from one another, okay? And so, and have different characteristics, right? And so that's part of this crossing over phenomena there. All right, so let's start off with the phases here of meiosis. I just went over this. My daughter was learning this in her biology class, so I had to go through this with her, right? And they learn it actually at a little bit more of a detailed level than what I'm going to be teaching you guys, because we got more important stuff to get to. If you want to do this stuff, take genetics class. So PMAT, remember PMAT, okay? So it's the order of the phases that we're going to be discussing here in a moment, right? So it's going to be prophase, metaphase, anaphase, and telophase. But here, we're going to start off in the interphase. So this is right before we are going to have our cell division. And so before the cell division, we've got to get the house ready. Okay, we're going to have some company over or something's going to happen. So we have to get everything ready. So the first thing we're going to do is replicate all of our DNA. Because remember, we're going to split off a cell, right? So we're going to split a cell, one cell into two cells. And then that would be the first part of meiosis. And then the second part of meiosis, we're going to split those two cells. They'll split also. So we're going to wind up with four. Remember, we're going to wind up with four daughter cells at the end of this process here. So we start with one cell. It divides, then we have two cells, and then those two newly formed cells, they'll divide again, and so we'll wind up with four cells there. So in interphase here, we're going to replicate all the DNA in the cell. And so now, instead of what we call, all right, a chromosome, now we're going to describe it as sister chromatids. Right? Those sister chromatids now, and it almost looks like an X configuration. And we'll talk about how when they do the whole lining up thing. But understand that we have just replicated, right, that DNA there. And so the sister chromatids at the center will have this structure here called a centromere, not to be confused with the centrosome. Y'all remember what a centrosome is? They're involved in cell division. Or not centrosome, excuse me, centrioles. Sorry, centrioles. You remember those two, like, yeah, those two cylinder things? They look like a toilet bowl, toilet bowl. They don't look like a toilet bowl. They look like a toilet roll, all right? And so normally when the cell's not dividing, they're perpendicular to one another. And then when they're about to divide, they go to opposite ends. I'll get into it, but they go to the opposite ends of the cell, and then they shoot out their mitotic spindle, and they're going to rip the cell apart, right? So anyways, the centromere is that joining there of the two sister chromatids there. Okay. So let's get into our meiosis 1, because you go through meiosis twice, meiosis 1, meiosis 2. All right, so now we have just replicated our DNA. So what we're going to do is we're going to split, all right, those copies here, essentially. So once we get through all of that, then we're going to pretty much do the same thing again, but there's some subtle differences in the steps there. So in meiosis 1, the primary thing that we're going to have is one cell that's going to divide into two cells. Right? And then during that first part, we're going to get some crossing over of the material there. Okay. So the homologous pairs, remember, those are the same chromosomes there. They're going to get split apart into our two new cells there. So those homologous pairs are going to get split apart, and they're going to wind up into those two newly formed cells. Still, we're going to have a haploid number. That's 23 chromosomes there. And so we're going to talk about those four phases, PMAT. Okay, so let's start off on the first one. I'll go through pictures here. So prophase 1, right? What you're going to see is we're going to pair up on the equator of the cell the homologous pairs of chromosomes because we replicated it. Let me show you quick pictures so you know what I'm talking about. All right, so here we have our homologous pair. One is from mom, that's the red. One is from dad. And so we just replicated them, and we're going to start to pair them up with one another. So again, a sister chromatid is going to be, right, that kind of X formation that you're seeing there. So we just kind of gather them up. We're getting ready for, all right, our replication. So it's going to look kind of like two X's hanging out near each other. So one's from mom and one's from dad. This is where you're going to see that crossing over phenomenon. And this is only going to happen in meiosis 1. It does not happen in meiosis 2. That's one of the differences there that you want to understand between meiosis 1 and meiosis 2. So that those two, you can see it on this picture here. You can, well, I'll zoom in. You can see it. See how there's a little bit of red here and a little bit of blue here. So they've literally, this little arm, whatever you want to call it, extended over and swapped, and this one extended over and it swapped out material randomly. It's a complete randomization process there. But again, it lends to that genetic diversity there. So you get a crossing over, and then remember, where's our DNA normally stored? Y'all remember? Where's the DNA? In the nucleus. So we got to get it out of the nucleus. So the nuclear envelope just melts away. So then they can just swim about, or whatever you want to call it, move into the cytoplasm of the cell there. Because now what we're going to do is we're going to enter into the second phase here, right? Metaphase 1. And this is where those tetrads, those two X's that are next to each other, they're going to just start to line themselves up on the equator of the cell. Come right back, promise you. So here we go. We can start to see our tetrad, our two X's, they're going to line themselves up onto the equator of the cell. And again, here are your centrioles on either side of the cell, and they're starting to extend their mitotic spindle here, because that's going to be that part of the cytoskeleton that's going to tear this cell apart. It's going to rip it right down the midline there. So how they arrange themselves, there's no known rationale. Go ahead.
I just heard about this, but some people having 45, 47.
Yes. Nondisjunction. Yes. Yeah. We're going to talk about that at the end of this. Yep. Great question. So here again, you're going to see those tetrads randomly place themselves. It doesn't matter. It's like some maternal chromosomes will go on the left side of the equator. Some maternal chromosomes will go on the right side of the equator. So they just plop themselves into position, and then the mitotic spindle starts to form, and we're going to start to see the beginning of where the cell will start to pull it apart. And that's where anaphase 1 comes into play here. So we're starting to pull the cell, rip it in two, and then we call that reduction division, because you're essentially reducing the number of chromosomes that were used to be in one cell, which was 46, and now you're going to wind up with 23 in each of those newly formed cells there. So that's why we call it reduction division there. So now each cell, all right, will have half the number. It'll be haploid, but it's still going to have those two sister chromatids. And again, what is it? What's that mean? The sister chromatids, that's that X. So a chromatid is one part of that X. So if I were to pull the X apart, you'd have one sister chromatid over here and one sister chromatid over there. Okay, so you're still going to have those two sister chromatids, which are going to be, right, identical, not exactly identical, because we've had that crossing over there, but you're still going to have, right, half of the DNA, all right, on this, on these two sister chromatids. One comes from mom, and some of it comes from dad, and vice versa on the other one. Yes, sir.
When you go to Ovarian, I just asked, but somebody has a disease and passed on to the child.
Yeah, you're talking about, hereditary diseases and whatnot. We're not going to talk about that too much. We're just going to do the non-disjunction, because we're not going to spend too much time, because I got to get into the female anatomy here. All right. So last phase here of meiosis 1, which is called telophase 1, PMAT. All right. Here again, we've pulled the cell apart. We're going to bundle up the chromosomes and the DNA material back into a new nucleus there. And then we're going to see 23 chromosomes in each cell there. And so at this point, for the rest of this part of meiosis 1, we're going to increase the cytoplasm. We're going to generate more cell organelles here. We're, again, because we just created 2 new cells, right? They're going to be pretty much almost half the size of the original cell. So we're going to create some of that material that was shared by both of those, by the old cell, and now we're going to replace and replenish what we just created there. So at the beginning of meiosis 2, this is basically what we're going to do. We have that X structure, okay, those 2 sister chromatids, and then what we're going to do is we're going to pull them apart and put one in one cell and one in the other. That's essentially what we're doing. All right. Again, I'm not going to get into too much detail, but I want to keep this as simple as possible. All right. So here you can see, right, with meiosis 1, all right, prophase, we're lining up, or we're bringing together our replicated chromosomes and pairing them up, and then we're going to line them up in metaphase 1 along the equator. Then we're going to start to rip the cell apart, right? So we're going to bust up our 2 tetrads. So then our 2 new cells are going to have 2 sister chromatids in each of the nuclei here. Still a haploid number, okay? It's not 46 chromosomes, right? It is 23 chromosomes. So when we get into meiosis 2, right, each of those cells are going to undergo this type of division. And I'll go through it again. Very similar, except for a couple differences here. So with meiosis 2, we don't have crossing over here. We start off with those 2 daughter cells that have those 2 sister chromatids. Those 2 sister chromatids. And like I said, we're going to separate those 2 sister chromatids. So you'll have one chromatid in one cell and the other chromatid in the other. All right, so I'll go through it with you. All right, here we go. So prophase, remember PMAT, prophase, same thing or very similar to the prophase there. I don't know why that says mitosis. That should say meiosis there. All right. So we have to break down the nuclear envelope, spill out the chromosomes or the sister chromatids that were in that nuclear envelope. Again, great test question here. There is absolutely no crossing over occurring in prophase 2. No crossing over. So in prophase 2, very similar to what we saw before, we're going to start to move, right, the genetic material, the DNA, along the equator there of the cell. So you can see here the nuclear envelope breaks down. We're going to start to move our DNA here towards the equator. And then that will be metaphase 2, will be same thing. We line up on the equator there. The centrosomes there, right, are going to pull apart, or the centrioles are going to shoot out their mitotic spindle. The mitotic spindle is going to attach onto the centromere of those sister chromatids. And now we're going to pull our X apart. And so as that's happening, right, we are going to enter into anaphase 2 as we pull everything apart. Now half of the X is in one cell, half the X is in the other cell. Here. Starting to pull it apart, forming our cleavage furrow here right through the middle of the cell. And then the last part, all right, the telophase 2 is going to be, we're going to package everything back up, okay? And then we're going to refill the cells with more cytoplasm, with any cellular organelles there that have diminished there, and you're going to be at the end results of this whole process of meiosis 1 and meiosis 2, we're going to wind up with 4 haploid cells, meaning 23 chromosomes. Like what? Yeah, like Michael Jordan, right? Kobe Bryant, right? Wasn't Kobe Bryant number 23 also? Or he went to 7. He changed numbers. 24. Okay, okay. That's right. Thank you. Thank you. Yeah, wrong. Yeah, I remember. Yep. So that's why, and again, not going to really cover it too much. Your 22 autosomes and your one other chromosome is the sex chromosome, which is the X or Y determining, right, whether you're going to be male or female there. And so again, at the end of this, we're going to see these haploid cells, all right, and females are going to eventually give us the secondary oocyte, which is going to be commonly referred to as the egg. And finally, all right, the spermatids or the sperm in males. So that's meiosis 2. So Willie, this is what you were talking about, the non-disjunction conditions here. So there's a couple more popular ones, but you have trisomy, which is someone that has 47 chromosomes instead of 46 chromosomes, or you can have a monosomy where you can have 45 chromosomes there, right? But basically, when you have trisomy, you've got 3 copies of one chromosome. And then if you have monosomy, you'll have 1 copy of the chromosome here. The situation, what happens is during that separation part, when we're pulling those sister chromatids apart, either in meiosis 1 or meiosis 2 there, we unfortunately are unable to pull, all right, the chromosomes apart appropriately. And we wind up with these. The more common one that you're probably familiar with is trisomy 21 or Down syndrome here. Right. And so, believe it or not, when you get into the medical field, if some of you are going into the medical field and you take usually patients' histories, a lot of my female patients, one, you'd be surprised how many have miscarriages, right? And I do believe that a lot of miscarriages are usually the body has sussed out something that could be potentially wrong with the embryo there, and it auto aborts. All right, and this is usually what you'll see with these non-disjunction conditions. Most of them, all right, will in fact, right, auto abort, right? Some will die in utero, right? Those that are born, again, this common one here, right? You can even see some of the conditions that, all right, folks that have Down syndrome. A lot of times, the reason why these folks don't live too long is because of their heart issues. Although, someone just told me that there was a gentleman that that participated in an Ironman. Have y'all heard about this guy? You heard about that? It's insane to me. Guy did an Ironman. I have no idea how old he was, but if you're not familiar with an Ironman, I believe is it 3 or 5 mile swim? I think it's a 3-mile swim, 100-mile bike ride, and then a marathon. It's like the cherry on the sundae. So this guy did all that. Right? That's pretty impressive considering that this now obviously they're healthy enough to do that, but most of the time they have heart issues there, right? And plus the other issues where they'll have degraded muscle tone and whatnot. But the fact that they did it it's really impressive. But usually with trisomy 21 Down syndrome, it's usually because the oocyte doesn't have, right, the correct or disjointment there that goes on during the separation portion there. And so this is why they discourage women over the age of 45 from getting pregnant, because your chances from having somebody with Down syndrome goes from like 1 in 10,000 to it's 1 in 100, I think. It's a significant increase, or I guess you could say decrease there. So I mean, statistically wise, it increases quite highly. I mean, you're going to see, and again, it's not guaranteed, but anyways, okay. So let's jump into the female reproduction, and we'll talk about some of this stuff in lab. So I'm going to kind of move through this a little bit, but I want to go over, right, one, the ovaries are considered to be, right, the primary reproductive organ in the females. In the males, it was the testes. In the females, it's going to be the ovaries. And so the ovaries are going to be the organs that are going to house, right, the oocytes, the developing oocytes here. So we're going to go through some of the stages here of the oocytes. All right, so the most basic and primitive form, where they all start off, what we call the primordial follicles here. And so essentially what it looks like is just a big, well, I got a picture here. It looks like a big circle. I shouldn't say a big circle, but it looks like this, right? So you have, all right, the actual oocyte, and then you have, like, it looks like squamous cells that cover, all right, the oocyte, and that is referred to as the primordial follicles. And when you're born, right, you have, in women, 1.5 million, roughly, which is a lot. Okay, but here's the thing, by the time you reach puberty, more than half of those are gone. And we'll talk about that here in a second. All right, so when you're born, again, these primordial follicles are going to undergo mitosis. They're just going to divide. All right, so you'll start off with the parent cell, boom, they'll divide and you get you 2 daughter cells, the same type of thing. But once you get your primordial follicles, we're going to stop meiosis right away. It's like you get into meiosis and then you stop right before you're born. So as soon as these cells are formed, they jump into meiosis and then they get to the prophase and they stop right away. So they replicate the DNA and they stay there for quite some time. Okay, so keep that in mind. 1.5 million are present at birth. Then, right, they will start to progress into the primary follicle. So I want you to think of a follicle as this room. And then if there's only one person in the room, they'd be the oocyte. Okay, so how I've described it before in the past, a studio apartment is a one-room apartment. And so usually one person lives in the studio apartment. So that's what the follicle is. That's the apartment, and the person living there is the oocyte there. All right, so here in the primary follicles, now we're going to see a change going from squamous shaped cells on the periphery. Now they're going to turn and develop into more of cuboidal cells. And so these are the granulosa cells, and we'll talk about what they're going to start to produce estrogen at the appropriate time. That plays a huge role, all right, in the female reproductive tract there, right? But you still here have at the center, all right, our oocyte, the egg, and it's referred to as the primary oocyte. So it's still arrested in the first phase of meiosis. It's still in prophase 1. It hasn't progressed yet, right? But keep in mind now, those peripheral cells are going to be producing the estrogen, because that estrogen is going to get the uterus, the uterine lining. We'll talk about this later on. It's going to prepare the uterine lining. It's going to start, the uterine lining is going to start to develop and thicken and more glandular growth. And we're going to get neovascularization, which is more blood vessels will start to go in through what we call the endometrium. Okay, we'll talk about the different layers there, but it's called the functional layer. And the functional layer is the layer that gets sloughed off during menstruation. So it's going to build that up. It's got to get ready for implantation. Okay, if hopefully, right, that the oocyte is fertilized. Okay, so this other structure that immediately surrounds, this comes later into play. Unfortunately, we won't really talk about it until chapter 29. Okay, but you've got this structure, all right, or this layer, right, it's made up of glycoproteins that immediately surrounds the primary oocyte. And this is what the sperm has to battle through, right? When it's trying to fertilize the egg, it's literally got to move through these cells, all right, these glycoproteins, these molecules, right, in order to penetrate, right, so for fertilization to occur. If you've ever watched a video of a sperm, all right, making its way and trying to fertilize an egg, I mean, it's really pushing in there. And we talked about the flagellum last class and lab class there, and its purpose there to help propel the sperm into that area there. So the zona pellucida is that layer immediately surrounding the oocyte that sperm has to get through. Okay, so the next stage here for our for our follicles is what we call the secondary follicles. Now the secondary follicles still contain a primary oocyte, so you don't want to get confused here. So primary follicles and secondary follicles are going to contain the primary oocyte, which is still arrested, right, or stuck in meiosis 1, right, that prophase here. But now we're going to see a new layer of cells form, and those are the thecal cells. And so the thecal cells, right, are going to be the cells that are going to be very helpful, because we're going to start to see androgens being secreted. Right, so the thecal cells produce androgens. Now, if you all remember, what are androgens? Yeah, that's male sex hormones. Well, female doesn't want that, all right? You don't want that. Or you're going to start to get male characteristics, all right? You can have hirsutism and all that, which ain't nobody want that, all right? But point being is, these thecal cells produce the estrogen. And so we need, all right, some cells to help, excuse me, did I say estrogen? Produce the androgens. And then the granulosa cells, which are in close proximity to the thecal cells, convert those androgens into estrogen, which is going to help continue on with what we call follicular development. Okay, it's almost going to be autocrine, which is going to help spur this process on. And then it's also going to be endocrine, right, which means that the estrogen is going to travel somewhere else and help, like I said before, maintain and promote the development of the uterine lining there. All right. So this is what we're looking at with our secondary follicle. Keep in mind, the primary oocyte still stuck in prophase 1, hasn't done anything yet, nothing too exciting going on there. But what we've seen is, all right, in the follicle, we've now developed another layer of cells here, and those are going to be the the granulosa cells. They go from one cell layer, now there's several cell layers, and then our peripheral cells here on the outside, those are the thecal cells. So the thecal cells produce the androgen, the granulosa cells convert it to estrogen, which helps this whole maturation process go on with the follicles and also helps to assist the uterus in generating more of an appropriate uterine lining there for implantation. And just to point out too, here's that zona pellucida right out here. So it's that kind of tan layer on the outside there, and that's where the sperm is going to fight its way through. All right, so now we're going to start to get a little bit more interesting here because we're going to see, right, some more changes occurring in our follicles. So we're now going to start to see a space, a cavity start to fill or form. And that cavity is called the antrum. And it's going to fill up with some serous fluid there. And what's going to eventually happen is that serous fluid is going to increase and increase and increase. And it's eventually going to push, right, the oocyte to the periphery, which is good because now it's going to get it ready for that oocyte to be released into the fallopian tube or the uterine tube because we're going to get ready for fertilization possibly there. All right, so again, we increase that serous fluid as we get closer to ovulation on day 14 for most folks. And so that's going to eventually push that oocyte to the edge. Not too much is still happening here with the oocyte. We're still stuck in prophase. I'll let you know when we get into the next slide here in a second here. It's going to then move on to a different stage there of meiosis there. But now what we're going to see is we've got a group of, well, let me just show you the picture. All right, you can kind of see now, you've got this antrum forming, and it's filling up with fluid, which is eventually going to push, right, the oocyte up here against the edge, but you'll still have the zona pellucida surrounding the oocyte, and then you'll have this layer of cells going around it too, this whole structure here that you're kind of seeing. If you've ever seen, I don't know, some artist renditions of the sun, and it has its sun. This reminds me of it. It looks like rays coming off of the sun here. Well, they call it the cumulus of forest, which is more like a cloud, right? But the corona radiata, which I'll show you here. These cells here, these granulosa cells, and they kind of have like these little spikes coming off, and that's what reminds me of the sun, because you'll have sun rays coming off at different angles there. But point being is this structure here, right, this whole thing, right, that's called the cumulus of forest, and then the corona radiata is this outer layer here, right, of those granulosa cells there. All right, questions about that? Not terribly exciting, I know. But now it's going to get exciting because now we're going to get to our next stage here where we achieve our mature follicle, also known as a Graafian follicle, also known as a tertiary follicle. There's a couple different names for this here. All right. So this whole time, not much has really happened with the oocyte. Well, guess what? The oocyte now is going to resume meiosis, and it's going to transition into what we call a secondary oocyte. And it is going to finish meiosis 1 only to go on to meiosis 2, right? But stop in metaphase. And that's where it's going to stay. It's going to almost get to the end of meiosis, right? Or excuse me, meiosis 2, and then it stops right before all right, the last two phases before anaphase 2 and telophase 2. And it's going to stay there and hang out. So these mature follicles, graphene follicles, or tertiary follicles, whatever you want to call them, this is what happens once a month. This is going to be, and you can see here in this picture, how our hole, as the antrum is filling up with more serous fluid, it's going to push this thing further against the periphery here. And eventually, all right, when ovulation occurs, and we'll talk about how that all happens with the hormones and all that here in a little bit. All right, but once that happens, this follicle ruptures and this secondary oocyte is going to be ejected out into the fallopian tube, hopefully. All right. Questions about anything? We'll walk it through a little bit more. All right, so ovulation happens. It occurs. We've ejected the secondary oocyte. It's somewhere in the fallopian tube. It takes about 6 days for it to make its way down a fallopian tube into the uterus. Most commonly, that's where fertilization will occur, is in the fallopian tube. In the one of the regions or one of the zones is called the ampulla, and so it'll occur there, right? Most commonly. So we're not going to concern ourselves with the secondary oocyte. We're going to talk about our follicles. So we've ejected out the secondary oocyte, and now what's going to happen is that mature follicle is going to transition here into what we call the corpus luteum. And the corpus luteum is actually going to become, for a short period of time, an endocrine gland. This thing here is going to release both progesterone and estrogen. And it'll do that depending on if fertilization occurs or if it, well, it's going to do it regardless. But if fertilization occurs, this thing is going to be in operation for a couple of months, right? If fertilization doesn't occur, which I'll mention here in a second, then this thing will degrade and turn into scar tissue, which we call corpus albicans here in a second, all right? But keep in mind, this thing plays a huge role because, remember, we still have to not fix, but we still have to build up the uterine lining. It doesn't happen overnight. Right, we're increasing blood flow, we're increasing gland formation there. The actual tissue in itself is thickening, right, ready for implantation there. So if fertilization doesn't occur, the corpus luteum degrades here into the corpus albicans, and it just turns into scar tissue. And a lot of times, right, the actual ovary will resorb that scar tissue. Sometimes, and sometimes it doesn't, right? But that's pretty much, all right, the storyline of the follicles. So again, think of the follicle as a structure that is, one, going to help with the development of the oocyte. It helps to promote, all right, its maturation, all right, from that prophase 1, all right, and then it helps it to complete meiosis 1 and then enter into meiosis 2 and then arrest in meiosis in metaphase 2. And so we'll actually see how at one point then it becomes its own little endocrine gland there. All right, so that's it for the follicles. Now we're going to get into, all right, the ovarian cycle. So there's a couple different cycles that we're going to discuss here for the female anatomy, and the ovarian cycle is going to be the first. Then we'll talk about the menstrual cycle. So hold on to your seats, right? Some of us are pretty familiar with this stuff. Others aren't, but that's okay. It's a good journey for us all to go into. All right, so let's first describe oogenesis. Right, this is that process of maturation for that primary oocyte, all right, developing into the secondary oocyte, which we just talked about here. So remember, let's rewind a little bit. Remember, in those primordial cells, you're born with 1.5 million of those primordial cells, right? And then you're going to see at one point, right? I'll jump back in here. You are left after birth, right, up to puberty. You go from one point 5 million. I just think that's an incredible number. You go from 1.5 million to less than 500,000 to about 400,000. So you have about 400,000 follicles in the ovaries. So if you want to do math, you got 2 ovaries, about 200,000 in each ovary, right? Enough to last several lifetimes, right? Several lifetimes, right? But eventually you're not going to continue on, right, and keep ovulating until the day you die. Obviously, you're going to enter into menopause, which we'll talk about in this chapter, what happens with that. But let me jump back into what I was talking about. Okay, so those primordial cells are going to be made up, that's going to contain diploid cells. And what's a diploid cell? Yeah, you're going to have, all right, 46 chromosomes there. And so diploid cells, if they engage in cell division, you'll have mitosis, which means that parent cell is going to provide you with 2 daughter cells that have the same number of chromosomes. So they would also be diploid, right? But what we're going to see here is if there's no mitosis going on, when we enter into meiosis, those diploid cells are going to generate, all right, at the end of meiosis 1, 2 cells that are what? Is it up there? It's up there. Right? 2 cells that are haploid. Okay. And then you are born with those cells that are going to be stuck in prophase 1. And at that point, they're no longer called primordial. They are now referred to as primary oocytes. And then we'll see the development of, all right, the granulosa cells initially, once they develop, all right, into that cuboidal configuration, now they're a primary follicle. Okay. And so again, you will have 1.5 million at birth, and that 1.5 million are all stuck in prophase 1. You want to know those, at least you want to know that those primary follicles are stuck in prophase 1 at birth, and they stay that way until puberty, and that's when you get atresia. And that's where you lose not over half, all right, about a third, about 2/3 regress. Okay. Whew. You guys ready to talk about the ovarian cycle? I'll be honest with you, when I was learning about the ovarian cycle, I was completely lost when I first, but that's okay. I don't experience that all my life, but now we get to discuss, all right, the ovarian cycle here. All right, so let's talk about what happens here, all right, during the ovarian cycle, and we're talking about the menstrual cycle and whatnot. So the first term that you see up there is folliculogenesis, and this is going to start in puberty. And like the term says, all right, you are going to see the formation, all right, and the progression of certain tissues, depending on which one we're talking about, whether it's in the ovaries or if we're talking about the uterine cycle there, what's going to occur there. We're going to talk about both here, right? But it all has to do with the endocrine system. Okay, so we briefly discussed it back in chapter 17. So we got to rewind all the way back to the beginning of the semester here. So we are going to see, all right, the hypothalamus playing a role in this, the pituitary gland playing a role in this, all right, and then we're also going to see what's happening in the ovaries and in the uterus. So keep in mind all of these sites here that are going to play a role. So the hypothalamus is going to be producing the gonadotropin releasing hormones, GnRH. Okay, so if you recall, the hypothalamus releases regulatory hormones. So those regulatory hormones are going to tell other glands what to do. So the GnRH is going to tell the pituitary gland to release follicle stimulating hormone and luteinizing hormone. Right, so we'll start off here with the ovarian cycle. So we have 3 phases. Right, the follicular phase, ovulation, and then we have the luteal phase. And that luteal should sound familiar because remember the corpus luteum? What we saw once, well, we know it, well, I shouldn't assume. Hopefully, we'll know what happens during ovulation. What occurs in the follicle? Yes, the secondary oocyte is released. Okay, and then we know what happens to that mature or Graafian follicle. What does it transition into? the corpus luteum. So it turns into the corpus luteum, and then it's going to play a role, all right, in this whole cycle, which we'll talk about. So when we look into folliculogenesis, and again, I don't really care about the whole preantral stage and the antral stage and how many days that are occurring, all right, 290 versus the 65, but I really want you to understand that the antral stage is going to be, all right, regulated by those two hormones, follicle stimulating hormone and luteinizing hormone, and that's going to play a role on what's happening in our mature follicle and in the antral follicle. All right, so let's get into the actual details. So let's start off here with the follicular stage. Essentially, think of ovulation, and again, this is all based on a 28-day cycle, right? 28-day cycle. So let's set a reference point. Ovulation occurs in the middle of the cycle. So that would be what? Day what? 14. Good. Okay. So we've got basically the stuff that happens before ovulation and the stuff that happens after ovulation. So the stuff that happens before, that's the follicular phase here. So that'll be days 1 through 13 here. And so you're going to see, right, the pituitary gland is releasing luteinizing hormone and follicle stimulating hormone. Look at the name, follicle stimulating hormone. It tells you literally what it's doing. Okay, it is going to stimulate the follicle to mature and go through those phases that I just discussed with you. So roughly about 20 of the primordial follicles will start this developmental process. Normally, one finishes, okay? But 20, it's like a race, you know? Someone's got to come in first, and that's what we're going to see here, right? So around 20, it could be 10, it could be 18, whatever. So roughly about 20 of them start the process during the follicular stage, and they go through all those stages that I just talked to you about, and one actually finishes. Now, at this point too, keep in mind, right, we got the granulosa cells, and we talked about the granulosa cells release estrogen and what that effect is, but it also releases this hormone. Now, I know that some of you weren't here. I'm not giving anyone a hard time. Some of you weren't here on Thursday, but we talked about this hormone in males and what it does. And inhibit in males, right, decreases the production of sperm. All right, so we don't get our sperm counts too high. Well, in the female here, it is going to make sure that we're not developing 180 mature follicles. All right, its job, again, it's almost like that negative feedback loop there. It is going to decrease because it does the same thing in males. It does the same thing in females as it does in males. In males, right, it is going to inhibit luteinizing hormone. Right here in females, it is going to decrease that follicle stimulating hormone, so we're not getting more of the follicles involved in this whole process, so we keep it down to a minimum and prevent excessive follicular development. I gotta slow my roll. If I talk too fast, I'll stumble over my words. I'm sorry, y'all. I don't know why I did that. Pick it back up. Okay. Questions about that so far? I'm trying to keep it just real simple. Okay, so in the follicular phase here, those whatever primordial follicles that started to transition into primary follicles, all right, we start to see some of them fall away, and then we develop into the secondary follicles, but eventually, like I said, only one. It's like the Lord of the Rings, one ring to rule them all right? One follicle is going to mature. Right? And it's going to eventually produce either the tertiary follicle or the Graafian or mature follicle there, or as the book refers to it as the vesicular follicle. All right, so that's it. And at this point, that secondary oocyte is pushed off to the edge, because guess what's going to happen next? What's next? What's going to happen? It's pushed off to the edge and ready for, like blowing up a balloon. We're going to pop the balloon here, right? And so what we're going to see is we're going to actually get into, all right, ovulation there. So one of the things that you should understand about the follicular phase here, right, as that primary oocyte graduates and finishes meiosis 1 and then develops in, and then we enter into meiosis 2. Remember, at the end of meiosis 1, how many cells are we finished up with? You start with one cell, and you wind up with, well, at the end of meiosis 1 and 2, but at the end of meiosis 1, you end up with 2, right? So one of those is going to be the oocyte. The other one is going to be this junk thing called a polar body. Okay. So the polar body, and it's different in sperm. Okay. But with sperm, at the end of meiosis 1, you're going to have 2 functional sperm cells. But in female meiosis 1, it's going to be 1, I hate to say useful cell and 1 junk cell, but that's essentially what it is. You're going to have, all right, your oocytes that has the potential for fertilization, and then this polar body, which just is, like I said, has anyone ever seen the movie Twins with Arnold Schwarzenegger and Danny DeVito? That's what it reminds me of. Poor Danny DeVito. The polar body's Danny DeVito, all right? And I like Danny DeVito, don't get me wrong, all right? I think he's funny in what's that? It's Always Sunny in Philadelphia. I think he's hilarious in that. All right. So your second cell, all right, that as you go from that primary oocyte to the secondary oocyte is going to get all the good stuff there. That's the Arnold Schwarzenegger. That's the one that gets stuck in metaphase 2. This is the one that is going to be hopefully fertilized here. And if it doesn't, again, most of us know, right? There's a 24-hour period in during ovulation in which some folks like to refer to it as the female is fertile, right? Where the possibility of ovulation occurs. That's because the oocyte itself just degrades, right? So again, don't hold me to 24% or 24 hours, but it's roughly around there. So now we get to the midpoint, all right? Ovulation. This is day 14. Right, this is where that mature follicle is going to pop, burst, whatever you want to call it, and release that secondary oocyte, right, into into the follicular tube there, or fallopian tube, right, into the uterine tube. And what causes this is going to be, all right, the luteinizing hormone. So some of you might have heard of the LH spike, right? So if you're tracking hormone production, and you will see, and I'll show you a picture here shortly, right, where it shows how much follicle stimulating hormone is being released at a certain time during the ovarian cycle, and how much luteinizing hormone is being released during the ovarian cycle. You'll see it. Luteinizing hormones kind of got a little gradual increase, and then all of a sudden it just takes a huge spike. There it is. You see, if you look on the graph on that picture there, where are you? Right up here. The green, right? That's the luteinizing hormone. So you can see that represents, right, the ovulation period, and it just shoots up. And that LH surge is going to stimulate ovulation. That's going to cause the mature follicle to release the secondary oocyte there. And so, right, the oocyte's going to make its way down the fallopian tube. So what happens now, right? Staying in the ovaries, right? We get into the luteal phase, and this is going to make up the remaining portion of the ovarian cycle. This is going to make up, yes, ma'am. Well, when you say PMS, I understand what it represents, but are you talking about physical, emotional, psychological responses? Oh, wait, are you saying before ovulation or before menstruation? Okay. Well, yes. Well, then the answer, this would be around that time. You know, it's hard to really say because I know it varies for some folks, you know, depending on how long. But yes, this is where you're going to see, and I'll show you some of the hormone release titers here in a second. But I won't even get into. Yeah, I do have to be careful, you know, because no, I won't even go there. Okay, so luteal phase. Here's what's happening. We now will see that mature follicle degenerate or transition, I should say, into the corpus luteum, and now we have our very own endocrine gland. And so it is going to be, again, on the lateral, or not lateral, but on the latter end of the 28-day cycle here. And again, it is going to produce progesterone, and estrogen the whole time. And I know I keep repeating this, but I don't want you ever to forget this. It is going to build up that uterine lining there. Right? And again, if fertilization doesn't take place, this thing degrades away. And it's going to then degrade into our corpus albicans there. So that's what we're seeing here, right? Once fertilization does not occur, implantation does not happen on the uterine lining here, then the corpus luteum here starts to degrade. It'll start to decrease its production of progesterone. It'll start to decrease its production of estrogen. And then we'll slough off that outer layer of the endometrium, the functional layer here. That's going to be, all right, referred to as menses, or the menstrual cycle there. You might want to know this term here. The first menstrual cycle is called menarche or menarche, depending on how you like to pronounce it if you're from Canada. Okay. But this, again, those numbers are arbitrary, 11 to 12. All right. Some of you could be sitting here like, well, I had my first period earlier than that. Some folks will say I had it later than that. I have a good friend of mine. She's got one daughter in high school. The other daughter's in 7th grade, and they both started around the same time. I think I told you that. So I just found I found that to be very interesting. I want to show you this slide here, the oogenesis slide. This one here, I think, again, I'm not going to go through it because basically what's on the slide is what I've been talking about. But what I'm going to suggest to you is, if you kind of want to have, and this is a good kind of appreciation here, and what I like about it is it shows you what's going on, all right, with the follicle or the follicular development, and also the oocyte maturation at the same time. So if you want to know, hey, I know when Dr. Kaz said, all right, when the follicle transitioned into the secondary or primary follicle, what was going on here with the oocyte? And you can see which stage of development that it's in. And actually, when it actually finishes meiosis 1, and then we get our 2 cells at the end of meiosis 1, right, the secondary oocyte and the polar body there. So it gives you a nice little description. So just something if you want to look at, and then you can actually see, right, some micrograph pictures here of the follicles too. I find it's what figure 28.6. Something if you're interested in. Again, you don't have to. Now this one here, I recommend that you take a good long look at this one. At first when you look at it, it's like, oh man, there's a lot happening on the screen here, right? But it actually is showing you, all right, the stages here, or the progression of, one, what the follicle is doing, what the oocyte is doing, similar to what we saw on this slide here, right? But also what your hormones are doing, both LH, FSH, and estrogen and progesterone. And then you can see what the uterine lining is doing. I really like this slide here. All right, at first look, it's like, wow, there's a lot going on. And as we move through, you can kind of see like what I was saying before, right? As we get to, all right, ovulation here, midpoint on the ovarian cycle, you can see, all right, FSH and LH relatively steady, relatively stable. All right, then you can see where the LH spike occurs, and that's what's going to cause the release here of the secondary oocyte from the mature follicle. And then also what we're seeing here with both estrogen and progesterone. You'll see that estrogen kind of spikes first, right? And then progesterone kind of spikes a little bit later with a smaller estrogen spike there. And again, you can see what's going on. Look at that. There's the formation, right? We're in the luteal phase here of the ovarian cycle. There's the formation of the corpus luteum. Look at your progesterone and estrogen levels. They kind of increase again, all right? Prior to that, right, what was producing the majority of the estrogen was, you remember what produces the primordial? Yeah, the granulosa cells and the thecal cells, all right? And so you'll see primarily it's those cells, but once we have our corpus luteum in play, then boom, everything's good to go. Then you'll also notice when the corpus luteum, if no fertilization occurs, look at the corpus luteum, it degrades into the corpus albicans, and then look at your progesterone and your estrogen levels. They just drop off. And then you can look, all right, what's going on in the uterus here? Oh, Well, you'll notice, all right, that the uterine lining is no longer proliferating. It's no longer thickening and growing. Then it's starting to degrade. There's your menses there, right? So that's when the menstruation occurs, and then we just start the whole cycle over again. So there's a lot to go on, and I know, and again, and I always wish that you all have more time to kind of look over this, except it's at the end of the semester, and everyone's worried about tests and exams and all that, to really appreciate it, all right? I'm probably one of the few males that actually appreciate that stuff. Okay, anyways, all right. So let's talk about a few more things here, and then we'll take a break. All right, perimenopause. First of all, what is menopause? Menopause is the cessation, all right, of the menstrual cycle. All right, and so usually that has to do, it's usually mostly an age-related thing, but there could be certain conditions that can cause premature menopause. All right, but perimenopause is, we're about to get to menopause. So in some cases, I've had patients, and I was told this by a primary care doc, that you have to have 1 full year of no periods, all right, before you're considered to be in menopause. And I've had patients come in and said, I haven't had a period in 8 months. I said, has it been a year yet? Nope. I said, then we don't know, because there's been times, all right, where depending on what's going on in a person's life, right, you can have periods of time in which stress, right? Folks that are anorexic have lost a lot of weight. You'll see it quite often in athletes, Olympic athletes specifically, and they always talk about the female gymnasts there. And there's a lot of concern with that, because when you're messing with the menstrual period there, you're messing with the hormones, and the hormones help with bone density, and there's a higher likelihood and predilection for what we call pathological fractures. Stress fractures of that nature are always a concern for those types of athletes, but they increase significantly more, right, when we start to mess with the hormone release there. All right, so perimenopause, we're getting close to menopause, but we're not quite there yet. Menopause, it has to be 1 year, and we'll see it usually in the mid years of an individual's life. Okay, so 40s and 50s, sometimes it's later, sometimes it's a little bit earlier. Okay, but basically, we're no longer going to see the maturation of the follicles and the maturation of the oocytes there, right? And then, so therefore, if we're not having that occurring, then we're not going to be creating the hormones that are responsible to high levels to proliferate the functional layer of the endometrium there. And so you won't get that buildup of the uterine lining there. And so if you don't get the buildup of the uterine lining, you're not going to have the shedding of the uterine lining. And so therefore, the individual will no longer have menstrual periods there. Okay. So the big thing that I tell folks when you're looking at menopause is this: you're going to see a relatively big drop off in progesterone and estrogen. And so that's one of the things that you'll see with individuals that get a hysterectomy. And a lot of times what they try to do for hysterectomies, they try to preserve the ovaries, because it's the ovaries that are producing or going to be releasing the estrogen and the progesterone there. So, okay. I got to stop here because I can't start this and then stop because this stuff is so good. I am going to send you out a video. Yay, I know. You're welcome. I'm going to send you out a video on this, and that's only so